Thanks very much, Wolfgang, for taking the time to speak to me today. And please, could you describe your role at the Manchester University?
Yeah, so my employer is Cancer Research UK, but so we are a cancer research institute, but we are embedded in the University of Manchester, but we're sort of a semi-autonomous institution. So we are funded predominantly by, we're a charity, we're funded predominantly by donations, by legacies, by charity events, but also obviously by some university funding and funding streams that universities are open to.
Yeah, that's great. Thank you very much. What are the main areas that you are currently working on, or your group is currently working on and focusing?
Yeah, so we are a service department. We cover a broad range of molecular biology aspects, and probably the biggest one by far is next generation sequencing. So we run a sequencing service for around 15 research groups. So these research groups are in a broader sense all involved in cancer research, and this ranges from basic research, research in model animals, tissue culture cells, but also preclinical work. So we don't do any clinical sequencing. So we always stay on the research side of things. We are involved in preclinical research projects. So we would also get involved in sequencing, patient biopsy samples.
That's great. Thank you very much. And what are some of the major challenges that you and the group is facing at the moment?
Well, the challenges always remain the same. There's a huge range of applications that we are faced with, and to go with it, a huge demand. So there's always the question of timely response and deliveries. One of the challenges that we're tackling now is sort of producing seamless workflows. So that means we have tracing of samples, from the origin, that may be from cells or from model animals or from patient samples, and trace anything that's done to those from the origin up to the production of this data, such as the sequencing data, and then including the analysis. So obviously there are things like LIMS in place, but what we're trying to achieve now is getting much more comprehensive traceability of that. Mm-hmm. That's just one of the examples of the priorities that I undertake.
Mm-hmm. Yeah, that's great. Thank you very much. It sounds very interesting. So you're heavily involved in sequencings. Where do you think the field is heading right now? And if we compare it where it was 5 years ago, for example, what do you think? What are the main areas that evolved within sequencing?
Yeah, I mean, obviously sequencing has become quite a staple technology for most research areas. And I think what we find is that there was a reluctance of using sequencing data as a means for data analysis. It was always 5 years ago, it was seen as something very niche market. Whereas now, I think technologies and also the cost allow it to be sequencing as part of a research project, without having to find huge extra resources. So it will, sequencing is just one part of a research project, and it doesn't have to be a very complex thing on its own. So that's one area. Obviously, there are sort of certain developments that we look at where we see multi-omics analysis. So sequencing does not just mean DNA or RNA sequencing, but we want to see an integration of data coming from various strands such as DNA, RNA, chromatin states, the proteome state. So integration, I think, is the big word here.
Yeah, that's great. Thank you very much. So your presentation at the event is going to focus on the opportunities and challenges in the single-cell sequencing era. Would you be able to give us a little bit of insights as to what you will be covering within your talk?
Yeah, obviously, we're not method developers. We are the ones that use methods. The challenge for us is that there is a flood of new methodologies coming through all the time, and to bring these new technologies to the bench, it requires validation, it requires benchmarking, it requires testing, and also the acceptance by the researchers. So this is the challenge for us. We like to offer certain services to our users, but we obviously have to find the priorities and find the right approaches. So we can't pick 10 different methodologies. We have to, because it would not be practical, it would not be cost effective. We have to find the right ones for the users, and we have to accept that certain technologies are not applicable or not cost effective to use. So this is sort of I find a typical scenario for a service.
Great, thank you very much. And how do you think the field is going to evolve in the future?
Yeah, as I alluded to before, I think the multi-omics approach is something that we will embrace. There are already a number of applications and technologies available, and I think this is only going to get more widely used. So we do RNA sequencing, we can do chromatin sequencing, or at the same time we can do protein analysis in parallel with RNA sequencing. There is an emerging field of spatial transcriptomics that's really, I think, only just at the beginning. So that would combine the histological information, or the spatial information of cells. It will combine that with transcriptomics, for instance. So this is something really exciting. So I think this is one of the areas of great development in the next few years.
Great, thank you very much. A lot of other people as well highlighted that the spatial biology, spatial area is going to gain more and more importance in the future. And I have one final question, Wolfgang. What would you like to achieve at our Congress this December?
Yeah, what I've found in the past is that once you talk about your own challenges and trials, you are met with quite a number of like-minded people. So basically, it looks like we're all in the same boat. Everyone faces the same difficulties, the same challenges, the same experiences, and it's really reassuring that to find out that what you're doing is kind of you face the same experiences that other people experience as well, and exchanging those experiences and ideas, I think is that's kind of the essence of what a meeting should be, shouldn't it?